The bioequivalence studies are required to be performed for the formulations to demonstrate the bioequivalence between the reference formulation and the generic formulation. Also, when there is a change in the existing formulation, that time also the bioequivalence studies are required to be performed. Bioequivalence studies can demonstrate that the formulation is safe and effective as per the reference formulation or in comparison to the reference formulation, and also in comparison to the formulation before any changes are done or before the SUPAC changes are done. A biowaiver means you can skip the in vivo bioequivalence studies. A bioequivalence study can be waived by the biowaivers. A biowaiver allows pharmaceutical companies, the pharmaceutical product applicants to skip in vivo B studies when strong scientific data shows that the product will behave the same as the reference formulation or reference product inside the body. So you cannot get the biowaiver directly. You need to provide the scientific rationale and the data so that you can support the biowaiver. So instead of testing the products in humans, a biowaiver relies on laboratory tests such as dissolution. Dissolution testing is a performance testing and it can give you support to demonstrate the bioequivalency or in vitro bioequivalence to support the biowaiver. This helps to reduce development time, cost, and avoid unnecessary clinical exposure while ensuring the patient safety. Now coming to the understanding about why biowaiver is important. Biowaiver plays a key role in the development of formulations like oral solid formulations and some other formulations like oral liquids and topicals. Biowaiver is not only applicable to the oral formulations, but it is also applicable to other route of administration. The biowaivers can help to speed up the product development and approvals because the bioequivalence studies are time consuming and costly. These reduce cost by avoiding the clinical B studies, support the life cycle management like reformulations, strength additions, and the site transfers, then SUPAC changes, site changes, and some others. These can maintain the patient safety by using strong in vitro evidence. The types are like BCS biowaiver, then additional strength biowaiver, then in vitro testing based biowaivers, and the SUPAC level biowaivers. BCS based biowaiver, the concept is if the formulation is containing the highly soluble drugs, which may be with high permeability or low permeability, the biowaivers can be given. So mainly the BCS based biowaivers are given to the BCS class 1 and 3 drugs. BCS class 1 are those which have high solubility and high permeability. BCS class 3 drugs are having high solubility, low permeability. These drugs are eligible if they show fast and very fast dissolution and if the excipients used in the formulation do not interfere with the absorption. Q1, Q2 similarity is required for the BCS based biowaivers. Generally, for the formulations containing BCS class 1 drugs, Q1 is required, and for class 3 drugs products, Q2 is also required. So to get the BCS based biowaiver, the drug formulation or drug product must dissolve more than 85% in specified time. For class 1, usually it is within 30 minutes. For class 3, it is within 15 minutes. Dissolution must be similar to the reference product across the different pH like pH 1.2, 4.5 and 6.8 and F2 similarity factor above 50, which can confirm that the test and reference formulations behave similarly and dissolve similarly. Then biowaiver for the additional strengths. So there are multiple strengths for the formulations. Like example, I have considered 5 mg, 10 mg, 20 mg tablets or capsules. These studies can be performed only on the one strength, whether it is higher strength, middle strength, or lower strength. Other strength can be viewed for the bioequivalent for the bioequivalent study, and you can get the bioequivalent if all the strengths have the same ingredients and similar quantities, that is Q1 and Q2. The manufacturing process is same for all the strengths. Drug shows linear pharmacokinetics and all the strengths show similar dissolution profiles. Then SUPAC, bioequivalent during scale up or SUPAC changes. SUPAC is a support for the changes to the approved formulations. SUPAC helps to avoid B studies when changes are made using manufacturing or during manufacturing using some excipient at lower or higher levels, such as changes to the batch size, site transfers, change in the manufacturing equipment or site. A bioerror is possible if the product composition remains similar within the SUPAC levels and dissolution is similar. Then the reformulation and life cycle management. Sometimes the formulations are required to be improved. For example, change in the coating material or tablet hardness. If the changes do not affect the dissolution or drug release, a bioequivalent may be applied. So this type of bioequivalents can support the continuous improvement of the formulation without repeating the B studies, and reformulation product improvement can be possible. ICH M9 global harmonization. ICH M9 provides globally harmonized guidance for BCS based bioequivalents. It aligns expectation of major regulatory agencies like US FDA, EMA, Health Canada, WHO, EMDA, ANVISA, and some others. This improves predictability, reduces the repeated studies, and it can support the faster global product development and the submissions. So whenever you come across the bioequivalent, you should think by which way the waiver can be granted. And understanding of the bioequivalent is required to all the pharmaceutical industry professionals, whether they are working in formulation research and in other departments like ADL, RA, and QA. Thank you for watching the video.